also from GATE 2022 Life Sciences Section P. Question number 31. Which one of the following compounds is not a translation inhibitor? Okay, and the options are A. Chloramphenicol, B. Cycloheximide, C. Puromycin, and D. Rifampicin. This is the NPTEL online course reference. So, we have various antibiotics, and these all antibiotics essentially target, these are the different targets. So, they essentially target the cell wall, right? They either target cell wall or protein synthesis, DNA topoisomerases, folic acid synthesis. They either damage the DNA or mRNA synthesis. Okay, these are the main targets they try to inhibit. So, few examples are very well known. So, the cell wall inhibitors we already know. We have class of penicillins, right? We have ampicillin, and this is the first antibiotic to be ever discovered. Then we have class of cephalosporins, and from there we have first, second, third generation cephalosporins. These generations are been derived because of antibiotic resistance. Okay, then we have various bactams and carbapenems. These are also derived, which essentially inhibit beta lactamase of the cell wall, right? And the cell wall which doesn't have any lactam, we have different glycopeptide inhibitions. So we have vancomycin, and these inhibits the glycopeptides. As you all know that the cell wall is essentially made up of carbohydrate and peptides, right? So we have either inhibition of the synthesis of this cell wall or the peptide inhibition, glycopeptide inhibition. Then we have protein synthesis inhibitors. These are also very robust. So in protein synthesis inhibitors, the inhibition could be either binding to 30S unit of ribosome or 50S unit of ribosome. Okay. So apart from this 38-50S, we should also not ignore, there are few inhibitors. They inhibit the tRNA binding, like tRNA translocation. Okay, we'll see one of that example here today. So they inhibit the translocation of the peptide chain onto the peptide synthesis, right? Then we have DNA topoisomerases, right? So these are also very common. So in E. coli we have different topoisomerases as that of us. So it is we can be able to easily inhibit this DNA topoisomerases. Main two classes are fluoroquinones and quinolones. These are also very effective antibiotics. Then we have folic acid synthesis. This is involved in the synthesis of folate, right? These are mainly sulfonamides and DHFR inhibitor. DHFR is a major enzyme, right? Dihydrofolate reductase is a major enzyme involved in synthesis of folic acid. So we have these inhibitors. This is also very important. There are few questions in GATE on DHFR inhibitors. Then we have inhibitors which carry out DNA damage. Okay. And then finally we have mRNA synthesis inhibitors. So in the question we had these 4 inhibitors. So I'll explain the action of all these 4 inhibitors. Then we'll decide which among them is not a translational inhibitor. So first we have chloramphenicol. So the action of chloramphenicol here is it binds to the 50S subunit of ribosome, right? So it binds tightly to the 50S subunit of ribosome, and as you can see, the site of binding is near to the peptidyl transferase site, right? We have here A site, P site, and E site. So A site is the site where the amino acid enters. Okay, it gets ready. P site is where the amino acid gets so we have a peptide bond formation. The amino acid gets transfers, and then we have a exit site where the uncharged tRNA whose amino acid is already transferred, the uncharged tRNA is excluded, right? So as you can see, chloramphenicol goes and binds the 50S ribosome exactly at the site where we have peptidyl transferase activity of this ribosome. So essentially what it does, it will inhibit the peptidyl transferase, right? It will inhibit the peptidyl transferase, and then we will have a truncated proteins. So it essentially targets bacteria. How? 50S ribosome is present in bacteria only, right? So it essentially targets bacteria, and we'll get truncated proteins, and bacteria will eventually die. Okay, cyclohexamide. So what cyclohexamide does? Cyclohexamide targets eukaryotics, right? Eukaryotic cell. So we have a 60S ribosome and 40S ribosome, and this is naturally occurring protein translation. As you can see, we have a AUG, we have a methionine here, the peptide tRNA with a methionine, it will come, then we'll have a next amino acid, right, and we'll have increasing number of amino acid polypeptide chain. What will happen in presence of cyclohexamide? It will come and block at the AUG site, right? AUG site in the interface of 60S and 40S, and it will lead to the truncation. It will lead to the truncation of the protein, and this is how it will inhibit the protein translation. Okay, it is targeting eukaryotic cells. We mostly use this in mammalian cell culture. This is very important. CHX, we use this in mammalian cell culture. Okay, just remember that it targets animal cells. Then we have puromycin. Okay, what puromycin is doing again is we have a polypeptide chain formation here. It has a amino group here. Okay, so this amino group attacks on the carboxyl end of this polypeptide chain, and it forms a peptide bond, kind of a peptide bond with the polypeptide chain, right? Because of this structure, the peptide bond, the truncated peptide is released, right? It is released, and we'll have a truncated protein again, right? So what puromycin does, it forms a peptide bond with the carbonyl end of the polypeptide, of the growing polypeptide chain, and we'll again have a truncated protein. And finally we have rifampicin. Okay, what rifampicin is doing is, here we have a RNA template and we have a RNA polymerase. So it is polymerizing this RNA polymerase, and this RNA polymerase is known as RNA dependent RNA polymerase. Why? Because we have a RNA template, this blue, and it is polymerizing, okay, on that RNA template. So it is RNA dependent RNA polymerase. It is adding new RNA. And what happens to rifampicin? Rifampicin has a aliphatic chain which distorts the shape of RNA polymerase. Essentially it is affecting the RNA polymerase, right? It is inhibiting the RNA polymerase directly, and it is not allowing RNA dependent RNA polymerase, right, activity. So we won't have any new RNA. So as we went through, chloramphenicol, it binds to the 50A subunit. Okay, so this is also inhibiting translation, right? Cyclohexamide, eukaryotic translational inhibitor, right? Peptide transferase activity in eukaryotic ribosomes. Puromycin is also a translation inhibitor, right? We have a truncated protein. It is bacterial, puromycin. And rifampicin. What is rifampicin? It is not a translational inhibitor, but it is RNA inhibitor. So what it is doing? It is inhibiting the RNA polymerase activity, right? So the correct answer is rifampicin.